Hello dear students, how are you? Today we will discuss translocation of crude sap. Please follow up with me in worksheet 9. In this video we are going to cover the following objectives. Deduce that in most plants the crude sap is transported by the conducting vessels. Identify the conducting vessels. Differentiate between absorption and translocation. So now let's start. Remember, in the last video we discussed absorption, and we said that absorption is the entrance of water and mineral salts to the plant through the absorbing hairs of the roots. These are the absorbing hairs, and through these absorbing hairs the water and mineral salts enter the plant. After their entry they become the crude sap. What is the fate of the crude sap found in the roots? What will happen to this crude sap? Where will the crude sap move to? Let's see. Watch the video, then answer the following question.
To carry out this experiment, you need a piece of celery including the leaves on the top, non-toxic dark food coloring, blue and red tend to work best, a glass plus a spoon, some water. First fill half of your glass with water, then add at least 5 drops of food coloring. The more you put, the better it is, and stir well. Now, cut the stem of the celery from below and put it in the colored water. Now you must wait a whole day, at least, to see how the colored water changes the celery. When you come back the next day, take a close look at the leaves at the top of the celery and at the bottom edge of the stem that was standing in the water. If you have put enough food coloring and have waited long enough, you will notice that some celery leaves have changed color to take on the color of the water, and that part of the cut bottom of the celery has also changed color. What do you think happened?
Now please answer the following questions. Where is the ink located? Where was the ink put in the video? Where did you see the ink? In the glass. Compare the color of the leaves before and after dipping in the ink. What was the color of the leaves before dipping in the ink? What happened to this color? Did it remain the same or did it change? The answer is the leaves are green before dipping in ink while they turn red after dipping in ink. In which part of the plant does the ink circulate to reach the leaves? We said before that a plant is composed of 4 parts: the stem, the roots, the leaves, and the flower. Which of these parts allow the passage of the ink? What do you think? In the stem. Indicate the path followed by the colored liquid. What is the path followed by the ink? How did the ink transfer from one part to another in the plant? It was in the glass. Then it transferred to the stem, and after that to the leaves. Now please open page 35 of the biology book and observe document E. Based on the observation, complete the table below. In this table we are talking about flower A and flower B. What is the manipulated variable and what is the result for each? Observe the figure. What is the manipulated variable? What is the difference between flower A and flower B? What was added only to one flower and not added to the other? We have what? The presence or absence of ink, since ink was added only to one flower. So in case A we are going to say the presence of ink, while for B we will say the absence of ink. What was the result of flower A in the presence of the ink? Did the color remain white or did it change? Change the color of the flower. You can see that clearly in the figure. What happened to flower B? Did the color change? No change in the color. Question number 2a. Compare the aspect of petals of the flower before and after dipping in blue ink. What was the color of the petals before dipping in blue ink? And what happened to the color? Observe here in the table for plant A. What was the color before dipping in a blue ink, and what happened to the color? So it was white. Then what is the new color? It is blue. So we will say the flower's petals were white before dipping in a blue ink. Why they became blue after dipping in a blue ink? Indicate the path followed by the colored liquid. How did the colored liquid transfer inside the flower? It transferred from the stem to the leaves and the flower's petals. What do we call the setup B? What is its role? What is the importance of the presence of a flower in the absence of ink? Why was flower B put in the absence of ink? Why? What do we call it? The setup that has no manipulated variable, or the setup that is not manipulated, what is it called? It is the controlled setup. What is its role? Why do we put a controlled setup in our experiment? In order to compare the results. So setup B is the controlled setup, and it allows the comparison between the results at the end of the experiment, and therefore indicates the role of the manipulated variable. Through which structure does the crude sap circulate in the plant? To answer this question, we made cross-sections and longitudinal sections of the different parts of the plant. To know what a cross-section and a longitudinal section are, watch the following video. Here, pay attention. A transverse section is the same as a cross-section. Here are some examples. Here we have cross-sections of a carrot and a cross-section of an onion. And this is a longitudinal section of the carrot, and this is the longitudinal section for the onion. Cross-sections and longitudinal sections of different parts of the plant. Here, this is a cross-section, as you see, when we make a cross-section and observe it under the microscope. Here is a longitudinal section. Here in these sections, you can see clearly the conducting vessels. So, what is the shape or the aspect of the conducting vessels? When we saw them, how can you describe these conducting vessels? They are in the shape of what? They are in the form of tubes. Where do we find them? Where can we see the conducting vessels? Where do we find them? In the roots, leaves, stem, and flowers. They are found in all parts of the plant. Indicate the role of conducting vessels. The role means the function. So what is the function of the conducting vessels? What is the importance of the conducting vessels in the plant? What do they transfer? The conducting vessels ensure the circulation of the crude sap from the roots through the stem to reach the leaves. So the conducting vessels allow the transfer of the crude sap from the roots to the leaves through the stem. What do we call the phenomenon of passage and transport of crude sap from absorbing hairs to leaves? What is this process called? The transfer of crude sap from the absorbing hairs to the leaves, what is it called? It is called translocation. So translocation is the transfer of crude sap from the absorbing hairs to the leaves through the conducting vessels. Evaluation. Number 1. Name the labeled part of the figure. What does this part represent? Conducting vessels. What is the aspect of this part? How do we see it? Observe it. What is the shape of the conducting vessels? In the form of tubes. Indicate the phenomenon taking place through this structure. What process happens through the structure? Translocation. In this figure you can see the entrance of water and mineral salts through the absorbing hairs of the root to the plant, which is called absorption. And you can also see the transfer of the crude sap from the roots to the leaves in a process called translocation. Here, as you see, these are the absorbing hairs of the roots that absorb water and mineral salts. Then the crude sap is transferred through the conducting vessels to the leaves. Here, these are the conducting vessels. Conducting vessels are tubes found in all parts of the plant. Objective 4. Differentiate between absorption and translocation. Absorption is a process that takes place through the absorbing hairs, while translocation takes place through the conducting vessels of the plant. In absorption, water and mineral salts are absorbed from the soil, while in translocation, the crude sap is transferred from the roots to the other parts of the plant. So here as a summary we can say, chlorophyll plants absorb water and mineral salts from their environment. These elements are absorbed at the level of the roots by the root hairs. Water and mineral salts form the crude sap that circulates through the conducting vessels from the roots to the stem, then to the leaves. As a homework, please underline and study number 3, 4, and 5 in worksheet 9 and answer the evaluation in worksheet 9. Do the experiments observed in this video. Thank you for watching and good luck.